Very good morning, dear students. We have been discussing the major concepts of enzymatic reactions and the mechanism of enzyme action. The previous class we have discussed how the enzyme activity is getting regulated, and in that aspect one of the very important concept for the regulation of enzymatic activity are enzyme inhibitors, and a specific type of inhibition process is known as allosteric inhibition. So the next topic mentioned in your syllabus, enzyme inhibitors and allosteric inhibition. So we are going to discuss what is the mechanisms and types of enzyme inhibitions and what are the various principles of allosteric inhibition. So before we are going to discuss the details about the enzymatic reactions and its inhibition, we should know that what do you mean by enzyme inhibition. Okay, what do you mean by enzyme inhibition, and what is an enzyme inhibitor? Okay, so an inhibitor or enzyme inhibitor is a molecule that disrupts the normal reaction pathway between an enzyme and the substrate. And in the previous class we have seen the enzyme very specifically bind to the substrate very specifically bind to the pocket of the enzyme. That particular pocket is known as what? The active site. So once the substrate is bound with the enzyme, the activation energy is getting reduced and the complex, okay, and the process is getting that enzymatic reaction will be happening properly, okay, and that is the mechanism of inhibition enzymatic reaction. And an inhibitor is a molecule similar to a substrate, okay, that can bind to an active site and simply block the substrate from binding. Okay, so that is the what do you mean by an inhibitor? Inhibitor is a molecule similar to the substrate and it can bind to the active site and simply by blocking the substrate from the binding. Okay, so that is known as the enzyme inhibitor. So it is very essential to regulate the enzymatic activity by proper inhibition. That is known as that is the activity of enzyme inhibitor. So the process of the inhibition by an inhibitor of an enzymatic pathway is known as enzyme inhibition. And based on the mechanism of action, the enzyme inhibitors can be broadly classified into two heads. Okay, based on the mechanism of action, the enzyme inhibitors can be broadly classified into two heads. One is known as competitive inhibitors, another one is non-competitive inhibitors. Okay, so we will discuss what is these two types of mode of inhibition. And another important aspect for the regulation of enzymatic activity is enzyme, that is regulatory molecules. Okay, these regulatory molecules, those molecules, they up or down the enzymatic activity by either activating or inhibiting the process. So that this molecule will be either activator or a inhibitor molecule that specifically bind to the enzymes. Okay, another important thing is known as cofactor. Okay, many enzymes are only active when they bound to a non-protein helper molecule known as cofactor. So we are knowing that the complete enzyme, both the protein part and non-protein part, is known as the holoenzyme. the protein part of the enzyme is known as apoenzyme, and the non-protein part of the enzyme is known as a cofactor or prosthetic group. Okay, so that is the important thing. And this enzyme inhibitors prevent the formation of an enzyme substrate complex and prevent the formation of the product. That is the main mechanism of how the enzymatic inhibition is happening. So enzyme inhibitors which block the formation of an enzyme substrate complex and thereby inhibit or prevent the formation of the product. And this inhibition of an enzyme may be either reversible or irreversible based on the specific effect of the enzyme, specific effect of the the inhibitor that is being used. Okay, the activity that can be reversible or irreversible based on the specific effect of the inhibitor that is being used. Okay. And in a normal enzymatic reaction, so we should, before we are talking about the regulatory aspects, you should know that how a normal enzyme is functioning. In a normal reaction, a substrate bind to an enzyme based on via an active site, okay, to form an enzyme substrate complex. Okay, in a normal enzymatic reaction, the substrate bind to an enzyme using an active site to form an enzyme substrate complex. So once the enzyme substrate complex is produced, the shape and property of the substrate and the active sites are complementary. Okay, generally this shape and properties of the substrate and the active sites are complementary, which result in the enzyme substrate specificity. Okay, which result in the enzyme substrate specificity. Okay, so when the binding happens, the active sites undergoes when the binding happens, the active sites undergo a conformational change to optimally interact with the substrate. Okay, that is known as the induced fit model. We have already discussed. Okay, the lock and key model and induced fit model. Okay, when binding happens, the active sites undergo conformational change to optimally interact with the substrate. Okay, so this conformational change destabilizes the chemical bond with the substrate, which will lower the activation energy. Okay, that is the main mechanism of a enzyme substrate reaction. Okay, as a consequence of enzymatic interaction, the substrate is converted into product at a very rapid or accelerated rate. Okay, as a consequence of enzymatic interaction, the substrate is converted into a product at an alarming pace. Okay, so that is the normal enzymatic activity. So this is what is the induced fit model in the normal enzymatic activity. This is proposed by Daniel Koshland. Okay, and that is the induced fit model. Both enzymes and substrate a dynamic conformational change upon binding. Okay, that is very, very important. The enzymes and substrate, they interact together and they undergo a dynamic conformational change upon binding. Okay, the enzyme contort the substrate into this transition state, thereby increasing the rate of the reaction. So as you can see in the picture, briefly illustrate, so what is the induced fit model. So here you can see the substrate entering to this is the enzyme, and here you can see two sides. One is this is known as the active site. Okay, this is the active site and this is the substrate, and here you can see the active sites and substrate, their specificity, they're more or less equal, but there is a little bit changes here. So when the substrate is coming and interacting, this specifically or complementary binding each other, this you can see this small distortion also that is leveled now. Okay, so now you can see the enzyme changes the shape slightly as the substrate bind. Okay, there is a shape change you can see. This is very not a perfect binding site, but you can see here it's a perfect binding site. So okay, that is enzyme substrate complex. So once it is happened, they bind the activation, they reduce the activation energy, the enzymes and product. Here you can see the enzymes that is there, the product is produced, and once it is produced, the product leaves the activation of the active sites of the enzymes. Okay, so this is known as the induced fit, and the interaction between this substrate and the enzymes is very specific, that is inducedly fit, that is undergo dynamic conformational change, and the interaction is mediated by several weak force. Okay, several weak forces such as van der Waals interaction, hydrophobic interactions, etc., and they produce the enzyme substrate complex, which lowers the activation energy, and that produce the products and leaves the product out of this particular pocket. So this is known as a induced fit model. So this is what I have told once again, whatever we have discussed here, the normal enzymatic reaction. Okay, this is what is the enzyme. Here you can see the substrate. Okay, here you can see the enzyme substrate binding. Okay, so what will happen? Then generally the enzyme that will be specifically bind to the binding side, the pocket. Okay, this is known as enzyme substrate complex. You can see the enzyme, the substrate is very specifically bind with the pocket, and there it is produces the enzyme substrate complex that reduces lowers the activation energy, and you can see the products are produced, and that is detached from the binding sites, and this product is produced by the activity of the enzyme. So this is what is the normal enzymatic reaction. Then what is happening when the inhibition is happening? Okay, how the enzyme inhibition is happening? I told you there are two types of inhibition. One is competitive inhibition, another one is non-competitive inhibition. Okay, so how a competitive inhibitor will work? Okay, so we can say a competitive inhibition involves a molecules other than the substrate bind to the enzyme's binding sites or active site, and the molecule that is other than the substrate, that is not their normal substrate, their closely resemblance with the structurally and chemically to the substrate, that particular molecule will come and bind. Okay, and that molecule is known as inhibitor. Okay, which are structurally and chemically related or similar to the substrate, and hence they are able to bind the active site. Since they are structurally and chemically they are same, actually they are able to go and bind with the active sites which are made for the substrate. Okay, so this competitive inhibitor block the activity of the active site and thus prevent the substrate binding. So I told you the specificity of this particular substrate, the binding pocket for the substrate and the molecule which are inhibitor, which are complementary, they are able to bind. Both the molecules are going to bind when you release this substrate and the inhibitor together. The inhibitor will rapidly go and bind the available binding pockets of the enzyme, so thereby occupies the binding pocket, and because of that reason the substrate will not have any space to go and bind with the same binding pocket. That means to say there's a competition between the inhibitor and the limited substrate. Okay, so there is a substrate. Then what will happen? As the inhibitor is in competitive with the substrate, these effects can be reduced by increasing the substrate concentration. That is why the substrate concentration is very, very important. If there are a lot of inhibitors in the system, the inhibitor will compete with the substrate and the inhibitor will go and bind, and they will be prevent the enzyme to bind the substrate. Okay, so how can you avoid this? You can increase the concentration of substrate. Only there will be little inhibitor and more number of enzyme. Obviously there will not be having any competition. The enzymes will be go and bind with the substrate, the binding pocket of the bind with the substrate will be going and binding with the binding sites of the enzymes, and they will be able to catalyze the biological process. So this is what I have told. This is what is the enzyme, and you are having the substrate and you are having the inhibitor. You can see they are structurally similar. The binding sites are almost structurally similar. And what will happen? When this both the substrate and the inhibitor releases to the system, what will happen? This inhibitor will go and bind with the binding pocket of the enzyme, and thereby there is no binding pocket for the substrate to bind. And what will happen? Since this inhibitor is already occupied the binding site, the enzyme will not, the substrate will not be able to bind. So the inhibitor bind to the active site and directly block the active site. There is no enzymatic reaction. This is known as the competitive inhibition, because there is a competition for the limited binding sites for the sites by the inhibitor and the substrate. That is known as competitive inhibitor. Then in the case of uncompetitive inhibitor, non-competitive inhibitor, there is a non-competitive inhibition involves a molecule binding to a site other than the active site. Okay, here also the binding is happening. The inhibitor will go and bind, but that binding is not happening to the binding site here. That is not the specific binding site. Okay, so the binding of the inhibitor to the that particular site, okay, that is known as allosteric site. I told you the binding will happen to other site. That site in which a non-competitive inhibitor will go and bind is known as allosteric site, which causes the conformational change to the enzyme's active site. This is an indirect method. Okay, so as a result of this change, the active sites and substrate no longer share specificity, meaning the substrate cannot bind. Okay, so it's very simple. We will discuss. As an inhibitor is not direct combination with the substrate, increasing substrate level cannot mitigate the inhibitor's effect. Okay, the previous case I told you, how will you prevent competitive inhibition? Simply increase the concentration, because the binding pockets are the same. But here the binding pockets are the inhibitor and the enzymes are not same. The inhibitor will be going and binding to allosteric site. The enzyme should go and bind it to the active site. Okay, so when you increase the substrate concentration, it cannot be prevented. The cannot be substrate level cannot be mitigated. The activity of the effect. So this is what I told. It's very simple. This is the enzyme. Okay, this is the substrate and this is an inhibitor. Okay, so what will happen? This inhibitor will go and bind specifically. First, this if you target both the substrate and inhibitor. Since inhibitor is a very small molecule, they will go rather than substrate. The size of the substrate always will be bigger than that of the smaller than that of the bigger than that of the inhibitor. The inhibitor will be having a smaller in size, and they will be go and bind first. Okay, so what will happen? This you can see the binding sites for substrate, they can go and bind, but inhibitor they can come on here. But what will happen if a non-competitive inhibitor like this one will go and bind? Then what will happen? This specifically will go and bind it to this binding pocket. It is very interesting, and see what is happening. Okay, there is an enzyme inhibitor complex, and due to this particular binding, it changes the binding pocket. Okay, so that the binding pockets are not complementary to this particular substrate. Okay, so the substrate cannot come and bind, because there is a specific shape you can see. This is like this and this is like this. Okay, they cannot go and bind. So the inhibitor bind to the allosteric site, the site other than the active site, and there will not be any reaction. Okay, which changes the shape of the active site. Okay, which changes the shape of active site. That is known as non-competitive inhibition. There is no competition for the single binding site. Okay, enzyme is having its binding site, and substrate is having, substrate is having its binding site, and inhibitor is having its binding sites for the enzymes. Okay, so here the inhibitor is going and binding, that block the binding of the substrate by changing the shape or conformation of the active site. Okay, that is known as non-competitive inhibition. So this is the effect of an enzyme inhibitor on the rate of reaction substrate concentration. I told you. So what will happen? The rate of the reaction. This is a normal enzymatic reaction that is proposed by Michaelis-Menten and Michaelis and Menten kinetics. Okay, and the substrate concentration increases, the rate of the reaction is also increases. When it is reaches the saturation level, slowly it will decrease. Okay. But in the case of a competitive inhibitor, the substrate concentration, I told you, when the competition is there, then what will happen? The rate is very slow. Then slowly what will happen when the substrate, if you increase further, as I said, then if you increase the substrate concentration, rate will be going and binding, but all the bindings is already over, then it will go like this. And a non-competitive inhibitor, when the interaction started, substrate concentration started, then the rate is also increases, but the inhibitor go and bind to the active site, it block the enzyme to come and bind, okay, to the active site, and it will go and it will specifically, it will non-competitive, it will specifically, it will go, and here you can see there is no increase further. So this is the graph of non-competitive inhibitor, this is the graph of competitive inhibitor, this is the graph of normal enzymatic reactions, okay. And what are the important, the comparison between the competitive as well as non-competitive inhibitors, okay. So very brief thing, in the case of competitive inhibitor, it compete with the substrate, okay, that's what I have discussed. Competitive inhibitor always compete, okay, that's a competition between the substrate and the inhibitor at the active site, and therefore which increases the Km value, okay, which increases the Km, actually is constant, okay. Then, and the Vmax is unchanged because with the enough substrate concentration, the reaction can still complete, okay. So Vmax will not be changed, but the Km can change, Km will increase, okay. So the graph plots of the enzyme activity against substrate concentration will be shifted to right due to the increase in the Km, while the Lineweaver-Burke plot will be sleeper when compared to the non-inhibitor. This we will discuss. Lineweaver-Burke plot we will discuss, okay. But in the case of conventional enzymatic activity, you can see it is going right, and it will be like this, almost similar to the normal enzymatic reaction. But in the case of non-competitive inhibitors, bind to another location of the enzyme, that is known as allosteric site, okay, that is decrease the V maximum, but Km is unchanged, okay. The V maximum, here you can see the V max is here, okay, but here V max is here, okay. You can see this is decrease the V max, but Km is unchanged, okay. So that is the thing. This is demonstrated by a lower maximum on the graph plotting the enzyme activity against the substrate concentration, and a high gamma intercept, okay, the high Y intercept on the Lineweaver plot when compared to the non-inhibitor, okay. This we will see actually, okay. And this is the competitive versus uncompetitive inhibitors, okay. Very, very important. Competitive inhibitor increase the V maximum, and V max will be increase K maximum, okay, Km, and V max will be unchanged. But in the case of non-competitive inhibitor, this decrease Vmax, Km will be unchanged, okay, just opposite, okay. Here you can see, and this is what I have told, the normal reaction, that's the competitive inhibition. This is the Lineweaver-Burke plot we have already discussed, okay. And here you can see, this is the inhibitor and this is the no-inhibitor, okay. This is the slope changes, Y-intercept same, okay. Competitive inhibitor, the Km is increased, okay. Here you can see the Km is increased and Vmax is unaffected, that is what I told. And the uncompetitive inhibitor, okay, here you can see there is no uncompetitive normal enzymatic reaction, that is uncompetitive inhibitor, okay. So this Km is reduced, Vmax is also reduced. Here you can see no inhibitor, okay, here it is a inhibitors, okay. In the case of non-competitive inhibitor, it's a mixed one, okay. This is Km will be unaffected and Vmax will be reduced, okay. So this is what is the, here Vmax will be unaffected, Km will be reduced. Here Km is unaffected and Vmax will be reduced, okay. What is the nature of the plot you can see, okay. So that is the competitive versus non-competitive inhibitor. So these are the factors where it is act. In the case of competitive inhibitor, it will go and bind in the active site. But in the case of non-competitive inhibitor, it will go on the alternative side, that is known as allosteric site, okay. So here the factor is the change of Km here, that is increase, okay. Here in the case of non-competitive inhibitor, that is unchanged. In the case of Vmax, okay, that is the maximum velocity, that here you can see, that is here competitive inhibition, that is unchanged, and non-competitive inhibition, that is decreased, okay. These are the three important features, the main comparison between the competitive and non-competitive inhibition. Then the next one we have already discussed, allosteric inhibition. So allosteric inhibition, if you use the inhibitor, the non-competitive mode of inhibition, if you use, that is known as a allosteric inhibition, okay. We will discuss once again. In a non-competitive inhibition, an inhibitor molecule bind to the enzyme in a location other than the active site, that you are clear, okay. In the case of an inhibitor molecule which bind to the enzyme location other than the active site, okay. This binding alter the overall shape of the enzyme such as it no longer bind with the substrate, okay. The binding alter the overall shape of the enzyme and it no longer bind to the substrate. Okay, this type of inhibition is known as allosteric inhibition. Most allosteric enzymes are made up of more than one polypeptide chains, meaning that they have more than one protein subunit. So many allosteric enzymes, they have a multiple protein subunit. Okay, so when an allosteric inhibitor binds to an enzyme, all active sites on the protein subunits are changed slightly such that they bind their substrate with very less effectively. Okay, when an allosteric inhibitor binds to an enzyme, all active sites on the protein subunits are changed, okay, such that they bind to their substrate with less effectively. There are allosteric activators and inhibitors. This type of inhibition, there are two types, and is using activators and inhibitors. The enzyme can regulate by other molecule that either increase or decrease their activity. Okay, so I told you there are activators or inhibitors. Okay, enzyme can regulate by other molecule that either increase or decrease the activity. The molecule that increase the activity of an enzyme is known as activator, while the molecule that decrease the activity of an enzyme called as inhibitors. Okay, so there are two types, allosteric inhibitors and allosteric activators. We have seen the role of allosteric inhibitors, and what is the role of allosteric activators? Okay, allosteric activators bind to the location on an enzyme away from the active site, including the conformational change that increase the affinity of an enzyme's active sites and its substrate. This we will be able to see in the next slide very clearly. Okay, the allosteric activators, they can bind to the location other than the active site, that is known as allosteric site, which changes the conformational, which increase the conformational, which changes the conformation, and due to that conformational change, the enzyme can come and bind with the substrate, and thereby it will happen. So this is known as the allosteric inhibition. Okay, this we have already seen. This is the inhibitors. What will happen? This is an active site, this is an enzyme. This is an allosteric site. Okay, enzyme has two sites. One is an active site, another one is an allosteric site. Where the allosteric inhibitor come and bind specifically, what will happen? This inhibitor will specifically go and bind. So the substrate cannot come and bind because they have changed the shape. Okay, they have altered the active site and the substrate cannot come and bind. This we have already discussed. This is known as allosteric inhibition. Okay, allosteric, the inhibitor will come and bind. Due to that particular reason, the active site of an enzyme will change its conformation. So you know that the active site is very specific for the substrate. Due to the change in the active site, the shape of the molecule has changed. Now the substrate cannot come and bind because the shape is specifically changed. Okay, and this is the allosteric inhibition. Then another interesting thing, allosteric activation. So what will happen? So this activators, so once sometimes the changed enzyme, so once it is changed, this will come on, this will go out. Then the activator come and bind. Okay, the allosteric activator come and bind where the allosteric side. So once it come and bind, what it will create? This will again change the conformation of the active site. Okay, this will change the conformation of active site, and that make the active site suitable for the binding of the substrate. Okay, the activator will come and bind here, and this will change the conformation of an active site so that the active site is now opened for the binding of the substrate. So what will happen? Then when the activator is bound, substrate is also bound, then there is an enzyme substrate complex formation is happening. That is what I told you. The activator will enhance, they increase the activity of enzyme and substrate interactions. Okay, I hope you understood. And this allosteric inhibition, the allosteric enzyme display a sigmoidal curve in contrast to the hyperbolic curve displayed in Michaelis-Menten equation. So this is what I have told actually. Okay, so here you can see, here we have seen that. Okay, this is the sigmoidal curve. Okay, in the case of an allosteric activator, a non-commutative inhibitor, and here it is a normal hyperbolic curve. So that is what we have a C. Okay. Then this is because most of the allosteric enzyme contain multiple subunit. I told you most of the allosteric enzymes are made up of multiple subunit which can affect each other when the substrate bind to the enzyme. The inhibition can either the K0.5 which is the substrate concentration for the half saturation, okay, the V maximum, or both. Okay, V max, you know that the concentration of the substrate required to operate half maximum velocity is known as the V max. That is known as K. Okay, so, this is the result in a shift of the curve to the right, and the case of reducing V max, shift the curve down. Okay, so this result in the shift of the curve to the right, and in the case of reducing V max and shift the curve down. Allosteric enzymes have two states, okay, a low affinity state that is known as T state, and a high affinity state that is known as a R state. Okay, allosteric enzymes have two states, low affinity states and high affinity state. The low affinity state is known as T state and high affinity state is known as R state. The inhibitors work generally they binding to the low affinity state of an allosteric enzymes, causing the enzymes to maintain its low affinity state. Okay, so the inhibitor work by binding to the T states of an allosteric enzyme, causing the enzyme to maintain its low affinity state. That is known as allosteric inhibition. And what is the importance of this allosteric inhibition? This is very much useful to study some of the mechanism like a feedback inhibition. We have already discussed in the previous class. Cymogen activation, feedback inhibition, we have already discussed. Okay, so this is extremely useful to limit the amount of an enzyme's product, as the product can go and inhibit the same type of enzyme to ensure the amount of the product is not excessive. Okay, this is the way how the inhibition is controlled. Okay, this is the way how the inhibition is controlled. This is known as a feedback inhibition. Okay, for example, ATP allosterically inhibit pyruvate kinase to prevent decreased formation of pyruvate, so less ATP is eventually formed. Okay, so you know that once the product is produced, okay, this product will go and bind to the initial step of the reaction, so that the process will not it will be inhibited. That means, example, ATP inhibit pyruvate kinase to the formation and prevent the formation of pyruvate, so less ATP is eventually produced. So phosphofructokinase with PFK, that is, you know that one of the important enzyme is allosterically inhibited by citrate, an intermediate of Krebs cycle. Okay, the Krebs cycle, you know that PFK is one of the important enzymes in glycolysis, and citrate, the citrate is the substrate, and that will be inhibited. That's an intermediate of Krebs cycle that will go and bind and inhibit phosphofructokinase. So this means that the glycolysis will be limited when there is high ATP generation from Krebs cycle. Okay, so this means glycolysis will be limited when there is high ATP generation from the Krebs cycle. So this is the importance of allosteric inhibition. That's all, dear students, of the enzymatic regulation. And the enzyme inhibition, once again we can glance through the points, what are the things we have studied. The enzyme inhibition, we have discussed what do you mean by enzyme inhibition, what is an enzyme inhibitor, we have discussed about the introductory concept, the regulatory molecules and cofactor we have discussed, then the normal enzymatic reaction, how it will happen, what is the mechanism of this particular normal enzymatic reaction, the lock and key hypothesis and the proposed and another model is induced fit model. The types of enzymatic reaction, there are two types, competitive and non-competitive inhibition. So the effects of enzyme inhibitors on the rate of reaction with substrate concentration we have discussed. Okay, that we have studied. Then competitive versus non-competitive inhibitors we have discussed. What do you mean by competitive versus non-competitive inhibitors? Then what do you mean by allosteric inhibition? Okay, we have discussed what do you mean by allosteric inhibition. Then we have discussed the mechanism of allosteric inhibition and what do you mean by allosteric inhibition. and what do you mean by allosteric activation? Okay, both inhibition and activation we have discussed. The next one we have discussed the importance of allosteric inhibition. Okay, what are the importance of allosteric inhibition? Okay, so these are the major points that is already discussed and already mentioned in this particular class. Okay, so that's all, dear students, and these are the major textbook you can study here. Okay, the main textbooks you can see, that is Principles of Biochemistry by Leninger. You can study that. Okay, then you can discuss the outline of biochemistry by Kohn and Stump. Okay, then you can study Voigt and Voigt. All this textbook you will be having all these details, and you can refer the other standard biochemistry textbook. Okay, so thank you, dear students, for watching this particular class. If you are having any clarification, you can ask me. So thank you very much.